This is our last portion for Chapter 8's lecture, and it should be short. So we're talking about regulation of enzymes, how we can actually regulate metabolism through the regulation of enzymes. And we've been talking about allosteric regulation, and one form of this type of regulation is cooperativity. And so cooperativity can actually amplify or increase enzyme activity. And the way that it does it is one substrate molecule will act as a primer to the enzyme so that the enzyme then is able or activated to more easily allow other substrates to bind to its active sites. So this is going to happen with enzymes that have more than one active site on them, which means that they're going to be quaternary enzymes or quaternary proteins that have multiple polypeptide chains with multiple active sites. So an example of this is hemoglobin that has 4 polypeptide chains that are able to bind 4 oxygen molecules. So when the first oxygen molecule binds to one of the active sites, it acts as a cooperative substrate because it then sets the entire hemoglobin molecule into a conformational form that allows easy binding for other oxygens to the other 3 sites. So this is a substrate is actually, in this case, oxygen acting as an activator. This slide here summarizes the different allosteric activators and inhibitors. So you can see here how an allosteric enzyme can have 4 subunits, which means it has 4 active sites. And in this case, when we have an allosteric activator, in this case, this is not a cooperativity because this is not substrate that's acting as the activator. Because what it's doing, instead of binding to the active site, it's binding to a different allosteric site, which is why it's called an allosteric activator. And so in this case, it sets the entire protein molecule, the entire enzyme, into a set structure that's able to easily bind the substrate. It's stabilized into an active form. In contrast, we can have allosteric inhibitors. And in this case, the allosteric molecule, again, is not binding to an active site. It's actually binding to an allosteric site, so a different site. And when it does that, it makes the enzyme go into its inactive form. So in this case, this enzyme has been inactivated or deactivated so that it can no longer function. Okay, and then the contrast to that is cooperativity, which is a type of allosteric activation. And the reason it's called allosteric is because the substrate's only binding to one site. It's not binding to all 4 of them. And so because it's doing that, it's actually causing there to be an activation of the enzyme by opening up all of the active sites. The last type of regulation of enzyme activity is through feedback inhibition. And so in feedback inhibition, in this case, the end product of the enzyme reaction, which is usually at the end of a metabolic pathway, so that means there have been a number of enzyme reactions, that actual product, whatever it is, will shut down the entire pathway. So feedback inhibition will prevent a cell from wasting chemical resources by synthesizing or making more product than it actually needs, because the product actually acts as a signal saying we have enough. So this next slide will show a diagram of how that actually works. Let me move this out of the way. So this is an example of isoleucine. which is an important biomolecule used in the body. And isoleucine is made through a series of steps. There are actually 5 steps in its synthesis. So what happens is threonine will actually bind to the enzyme. And when it does that in the active site, it's the substrate of this first step. When threonine binds to the site, it produces the first intermediate. So the enzyme here, enzyme 1, is threonine deaminase. And so it makes intermediate A. Then intermediate A combines with this enzyme 2 to make intermediate B. Intermediate B binds with enzyme 3, which makes intermediate C, which then binds with enzyme 4, and so on until we get to the end product, which is isoleucine. So this is, again, when we're talking about a metabolic pathway, has a number of steps in order to get to the final product. And with feedback inhibition now, this product then can go back up to our threonine deaminase and act as an allosteric inhibitor, because it will bind to an allosteric site to prevent this enzyme from any longer accepting threonine in the active site. So thereby it shuts down the entire pathway by stopping it cold at the first step. So this is again called feedback inhibition. And that is the end of this unit and this chapter 8. There's a lot of information in this unit, so go over your notes, go over the reading carefully, listen to this if it's helpful, and I will wish you good luck in your unit test this week.